Hello everyone and welcome to the INFLANET MOOC course. I'm Saatvik Potluri, a Marie Curie Research Fellow at CNRS, and in this MOOC course I will talk about high-resolution imaging and different techniques and setups used in high-resolution imaging. So to get it started, what is resolution? Resolution is the ability of the microscope to distinguish between details in a specimen, or simply put, it is the minimum distance between two different points observable by the observer. So the first thing we need to understand is why do we need high-resolution imaging? High-resolution imaging is a technique used in different fields like biology and cosmology to understand and observe different events that are unable to be observed by the naked eye. Either because these events are at too small of a scale, or if the events are too far away to observe. In this MOOC course, I will be talking more about the biological aspect of high-resolution imaging, as that is what we do in our lab, and the different methods we use to observe this. With high-resolution imaging, we are able to observe the close interaction between cells and the proteins they produce. We can also observe cellular movements through tissues or organisms in live once the setup has been optimized for the sample being imaged. The optimization of the setup is crucial to high-resolution imaging. Different proteins and tissue components can be visualized using various sample processing methods, employing antibodies or dyes. This would give us a clear idea of protein and cellular functions and how cellular mechanisms actually work. High-resolution imaging is also often used to observe the impact of drugs and chemicals on different models, either in vitro, like in human or mouse cells, or in vivo, like in zebrafish embryos or in mouse. The efficacy, potency, and function of these drugs could also be observed over time. With the various advances in science, many types of setups have been built and optimized in microscopes. Each of these setups have their own advantages and their disadvantages, and these advantages and disadvantages are considered before observing a specific process or an event. One of the most standard forms of microscopy used in labs is bright field microscopy. It is quite simple, with the illumination of the sample with light and using a small lens to observe the specimen more closely. Its simplicity and cost-effective methodology makes it quite frequently used in labs to observe the sample. Although specific components of the sample cannot be observed with it and the resolution with it is limited. Moving on from the bright field, fluorescence microscopy is also quite frequently used, with the key advantage of this microscopy being that we can observe specific parts of the sample and being able to label components individually with it. After this, light sheet microscopy is also a relatively new technique that has become popular in the field, also employing the use of lasers to illuminate a single portion of the sample and focusing on it to see the component. Confocal microscopy uses a fluorescence, but most importantly employs a pinhole to avoid background and noise from the illumination. The principle of confocal microscopy is modified with the addition of a spinning disc or many other alternatives to illuminate the sample only as much as it's required and observe this individually. After this, there is also multiphoton microscopy, which similarly employs a laser, but it excites the sample at a much higher wavelength to improve the penetration and get a greater Z resolution, while also limiting the toxicity of the laser. Super resolution microscopy is another technique that is used when the resolution of the sample and the image needs to be higher than the diffraction limit. While using super resolution microscopy, there might be disadvantages and sacrifices like factors with time. So to talk about these advanced forms of microscopy individually, light sheet microscopy employs the use of fluorescence lasers to illuminate the sample perpendicular to the direction of observation. The laser beam is transformed into a single plane of illumination while the objective is aligned and focused to this area of illumination. Through this way, illumination of the whole sample is avoided to prevent photobleaching or the fading of this fluorescence through time, and prevent phototoxicity, the damage of the sample through excessive illumination. These are two key factors that constantly need to be considered. Light sheet microscopy improves the speed of imaging as well, allowing us to image the sample at a much greater speed. The resolution of this microscopy is similar to a fluorescence or an epifluorescence microscopy and is dependent on the objective and wavelength used, as we will see later with the diffraction limit. This type of microscopy specifically also produces large amounts of data while having a few artifacts like the appearance of stripes on the image due to obstacles in the path of the light. Confocal microscopy is extensively used in industry and the scientific community to produce images with a high resolution and contrast. The main principle of confocal microscopy is the addition of a pinhole in front of the detector to reduce the noise and increase the signal-to-noise ratio. In confocal microscopy, only a single point of the sample is illuminated using a pinhole, which allows us to observe the excitation of the fluorophores only in this point. This redacts the noise. As the emitted light is reduced by employing the pinhole, it often requires a longer exposure time, increasing the time taken to capture the image, while also increasing the photobleaching and phototoxicity levels in this case. Confocal microscopy can be modified by the addition of different components. One commonly being used is the spinning disc, which is also known as a nip-cow disc. A spinning disc is a disc with a series of pinholes which is present in front of the laser to illuminate the sample separately in a pattern. This lowers the exposure time of the light on the whole sample and reduces the phototoxicity and photobleaching of the sample by only illuminating a specific portion. This is often used in live organisms and live cells to limit damage to the samples during live long-term imaging. Multiphoton microscopy also uses a similar principle of exciting fluorophores in the samples and detecting the output with the use of a detector. The main difference with multiphoton microscopy is that the excitation wavelength used to excite the fluorophore is much greater than the single photon excitation wavelength which is used in other microscopy. The fluorophore is excited at 2 or 3 times its actual excitation value. For example, the single photon excitation wavelength of GFP or green fluorescent protein is 488 nanometers, while in multiphoton or in 2-photon microscopy, we excite this fluorophore with 980 nanometers. By exciting it at a higher wavelength, usually within the red spectrum, we use less energy on the sample, limiting phototoxicity. This also allows us to image the sample deeper and get a greater resolution in depth compared to the confocal or light sheet microscopy. Multiphoton microscopy also has a specific advantage of being able to image specific cellular components like collagen and myosin by using the principle of second harmonic generation or third harmonic generation. This is due to the structure of the protein reflecting the excitation wavelength at half of its original wavelength. Multiphoton microscopy can be performed as 2-photon microscopy or 3-photon microscopy, limited by the excitation wavelength of the laser and the detection capabilities of the detectors. Finally, we will talk about super-resolution microscopy. To understand super-resolution microscopy, we should first mention the diffraction limit of optics. The Abe's diffraction limit states that the resolution of a microscope is limited by the wavelength at which the fluorophore is excited and the numerical aperture. The numerical aperture can be adjusted by the change in objectives, but the excitation wavelength is not changeable. Super-resolution microscopy breaks this Abe's limit by focusing light at a single point and scanning the point from different angles, point by point. To do this, this temporal aspect is often sacrificed to take multiple images which are reconstructed into one image with a high resolution. This is not achievable by normal optical setups. There are multiple types of super-resolution microscopy methods like STED and SURF present in this.